I'm Fiona Greer, and I'd like to welcome you again to the third in our series of short, informal roundtable discussions on the theme of biosimilar analytics, hosted by BioPharmaSpec. In the previous video, available on the BioPharmaSpec website, our two expert speakers, Dr Andrew Reason and Dr Ash Ramzan, had so much to say regarding knowledge-based biologics development that I was unable to ask them all the questions I had prepared. So I persuaded them to continue to answer the final two questions now. I'm pleased that we can continue our discussion on this issue, particularly on the issue that we started to discuss, the issue of robust and reliable methods for early characterization. On a more focused point then, Andrew, once you have a data package with the necessary supporting qualification data, how can this be transferred into supporting the clinical development phase?
Yes, good question, Fiona. So basically in our laboratories, a qualification is defined as an ICHQ2 validation, effectively. The only difference between what we do and something you would call a full validation is that we don't do it to GMP because we're in the early phase preclinical development part of the product development. So, but once you've qualified a method and, by the end of the preclinical phase, you've probably analyzed quite a few batches of material, there's a very good understanding of the product, how it behaves when you're analysing it, whether it's primary, secondary, or tertiary structure, and also which methods are critical to that product. Which methods are you using to make the big decisions? Which methods are effectively going to become release tests? So those methods generally we would be qualifying for the product itself. At BioPharmaSpec, every method we use is qualified, but it's qualified with standard peptides, sugars, glycoproteins, or what have you, to understand the basis of the method and the experimental error on that basis. But methods that are critical to that product and are going to end up being release tests, we would qualify specifically for that product. You can therefore set acceptance criteria and have a lot of knowledge for establishing that GMP validation study plan, protocol. And that's obviously a huge saving in time if you're going to be transferring that method into a GMP environment, and the knowledge that creates. So yes, I mean it's huge in terms of understanding the product, how it behaves in the methods, particularly the critical ones, and the passage is generally product qualified methods becoming release tests and then going through a GMP validation for release testing, into the clinic and onto the market eventually.
And from the clinic and the regulatory point of view, Ash, do you see that as a valid progression forward?
It's a continuum. So if you imagine when you are, as Andrew says, selecting the test that you're potentially going to be using, you start off with every single possible test and you build down into the applicable, the appropriate, those tests that gives you some useful information about characterization, all the way down to those tests that define the PQS, the product characteristics, the quality specification. And it's those tests that are usually expected to be fully validated, but they're expected to be validated prior to phase 3 clinical studies. Now, my view is that there's this grey area between validation and qualification. You can have qualification that's almost complete validation, or you can have validation that's you could interpret to be deficient. So the way we try to interpret this is, and because some of these tests are very, very complex, they are biological tests for biological molecules, both of which have intrinsic variability, so setting specifications can be really challenging. We look at fitness for purpose. So does the test give you some meaningful data that you can interpret in a value-adding way? And it comes back to looking at the holistic molecule again, rather than just picking one or two tests, because the complexity of these molecules doesn't lend itself to enabling one or two tests to say, we know everything about this molecule now. So we see it as a continuum, and You're continuing developing, adding, reinterpreting, and sometimes you might find a test that you thought worked really well 5 years ago, now doesn't.
Okay. And what would the reason be behind that? Would it be the method just hasn't moved on with the times? It's not the latest analytical method or the best analytical method?
It's usually because people like Andrew have developed better methods, which give you a much clearer picture of what you're looking at. So it comes into focus and you realize actually that you may have been looking at an artifact. And that's why we can't be close to developments. If you look at the developments in mass spec of the last 10, 20 years, they've been huge. And that's it. And that's when we talk about knowledge base and understanding how to interpret data, don't close off and say, well, this is it. Specifications can keep changing as methods keep changing.
Yes, well, I've seen that all throughout my career. When I came in 30 odd years ago, mass spectrometry was the new method on the block for protein characterization, and now where would we be without mass spectrometry? So all right, time for a final question then. Is structural characterization not needed during the clinical development phase then, Andrew? Perhaps you'd like to comment on this first. Do you finish before the clinical development phase?
Well, yeah, I was just thinking while Ash was speaking that one of the reasons why things change is a new manufacturing site might be installed for more material through clinical trials, etc. And it's very difficult for different manufacturing sites to produce exactly the same product. They may even be using different suppliers for excipients, and all of these things can have an impact. It does speak back to, testing everything during development to make sure what the critical parameters are, and then it should be relatively easy to get around these things. But, in the clinical phases, obviously, some of the qualified methods may be helping to make decisions until full validation is in place. And, the process may change. There may be a scale up, there may be another manufacturing site, and you are still then beholden to do a comparability study of that new product. You have to compare it with a reference batch and show that essentially that new manufacturing site or that small change in the process hasn't affected the product. And people say all the time that shouldn't happen, as you're approaching phase 3, but it happens all the time.
And I presume also you might assist with troubleshooting if...
Yeah, absolutely. Yeah, you're absolutely right. If there's an out of specification result or a new peak appears in a release test or whatever it might be, then there are generally characterization methods sitting behind that to be able to say quite immediately what the issue is, whether it's with the product or an excipient or whatever it might be. And we've had, lots of occasions where those sort of things have happened, from plasticizers leaching into products to reductant being present and structure changing and all sorts of things. Even when, you are at a phase where there is ultimate control, things still happen.
Yeah, okay, thank you. Ash, do you have any final comments on that?
I think I just echo what Andrew's just said. I think structural functional characterization will always be needed. I don't think I can't foresee any time where any regulator would be happy to dispense with that type of information, particularly for these types of molecules. And Andrew mentioned different sites and subtle differences in materials. Well, the climatic differences, changes, so even subtle differences can have a significant effect on these molecules. And of course, there are the big ticket items such as tech transfer, comparability that we all think about. But I think it's always useful to put yourself, if you can, in an imaginary kind of way, in the place of the molecule and see what the environment is doing to you. So I can't imagine a time when we will not have to report this kind of data. And the regulators are actually encouraging us to carry on doing that. I've been to a very recent FDA meeting where the reviewers there were encouraging us to, if you are generating data and you cannot interpret it, so you don't actually know what it means, share it with us anyway. They'd like to know. So you can see that they're hungry for this information as well, because they also recognize that, in the future, whilst we can't understand something now, it's likely that we'll understand what's going on at some point in the future.
There might be a key that transpires to unlock the solution to those data. Yes, okay, great. Well, once again, I'd like to thank you both for a stimulating discussion. Thank you all for listening, and to BiopharmaSpec for hosting. If you missed the first part of this conversation about knowledge-based biologics development, it's available on the BiopharmaSpec website. So please look out for our next event by visiting their website and following them on LinkedIn. Thank you once again.